So we're over to Owen Powell, who was also nicely introduced by Graham, so I'll let you just carry on.
Thanks, Christine. Yeah, I think there's been quite a thorough introduction to what I have to do, so hopefully that makes this presentation pretty easy. Okay, so Graham introduced this slide earlier and mentioned where he sat, and I think I'll just point this here to say that I'm talking about how can we bridge these different levels of biological organization into a predictive framework to help an end user, in this case a plant breeder or a breeding company. So we didn't plan this, but we had the same slide, but I've adjusted it for me. So the prediction can be used many different ways in a plant breeding program, and a lot of focus when we talk about plant breeding is the development of varieties, but that is also just one piece. So when I'm talking about prediction today, I'm talking about technologies that could help the overall breeding cycle that a plant breeder or a breeding team would perform. And that really focuses on the genetic gain, which people have mentioned. But what I've done here is just sort of pull out an equation which is often used by quantitative geneticists or breeders to think about how they can operate this whole process that Graham explained before. For prediction itself, in this figure, we've highlighted how prediction technologies can be used to leverage understanding from previous experimental data to inform decisions like crossing and generation of new genotypes. And this was a big advancement, and this is where a lot of these whole genome prediction technologies that were discussed before are being used. So critically, we want to understand how we can use prediction to move, wrong button, to move this reference population of genotypes, so this is our germplasm, forward for multiple traits simultaneously, which is the delta G, which connects right back here. And prediction can affect many different pieces of this equation, but today I'll just focus on the selection accuracy in a later case study. So my talk was better breeding with more biology. Nice and vague, can go many ways. But what I'm talking about is how do we move from prediction methodologies at the moment, which treat interactions as secondary pieces to go after, and what they're trying to do is capture these average sort of effects which persist over the broad spectrum of the data set you might be looking at, or the problem you're looking at. Not to undersell the work that's already done in the quantitative genetics and breeding space is that people do and have developed models to go after these interactions, but it's very statistically based, and it's trying to still go from the level of genomic information with these dots up to the complex traits such as yield. So there are already approaches that can try and tackle this. What we've been developing in the center that derives from a lot of the work that was presented earlier by Charlie is how do we try and capture our current understanding of interactions and networks of biology and explicitly incorporate this structure and understanding into prediction algorithms. So how do we include a lot of the information that's being generated in the center in between what breeders currently operate with in terms of genomic and phenotypic data? And on the bottom, I think this just highlights that it allows to explicitly get estimates for a lot of these multitude of interactions that could be going on from trait by trait through to environment and agronomic management. So if we could develop such a model, which I'm going to talk to you through now, could we deliver better prediction outcomes to breeders? That's what my work's been trying to ask. So how are we going to test this? So we're going to test it using the data set, this virtual data set that's been introduced before. And what this shows is it's a multi-environment trial from 2 sites in Queensland. So this is Dalby, just outside of Brisbane, on the map here, and up in central Queensland, and a couple of years that vary in their in-season rainfall. And as Graham already mentioned, this is a population where we have genetic variation for 3 drivers of grain yield in sorghum across the sorghum belt. And that we generated these outputs using this sort of genomic information coupled with the APSIM sorghum crop growth model, and this is what's generating the G by E, which you can see a slice of based on these sort of distribution plots, where we're changing the variation in grain yield and also the average performance. Looking at those distributions a little bit deeper, what you'll also see is that if we look at the relationship between grain yield across the different trials, is that we're seeing strong and positive to very moderate or moderate negative correlations. So this is causing the varieties, shall we say, of different plants to change in their ranking. So when a breeder wants to select the best, this is going to be a pretty challenging task for them to do so, if you're not thinking about what's generating the underlying interactions. And again, a plug for the data set. So to specifically test that, what we've been working on is first setting up this modeling framework that we call APSIM WGP. And in the simplest way I could describe it is we're taking what genomic prediction's doing, where we have a load of genomic data and we have a load of complex trait phenotypes such as grain yield, and they would try and map the relationship directly between those two sources of information. What we do is we drop the APSIM sorghum model in between, which captures a lot of these underlying developmental and growth processes in a mathematical framework to connect these two sort of pieces of information that sit at extreme ends of the biological hierarchy. And then we're going to train this on our data set and see what comes out. So this is sort of a result. So what I did was I actually trained each algorithm on each particular trial and then used the information we had from that trial to predict the remaining trials. So what you'll see is two colors of circles and an empty and a filled circle. So the empty circles are where we've sort of the training trial or the training environment, and the filled circles are when we're predicting into a trial that wasn't passed to the algorithm. And just the colors relate to whether it was the lower or the higher rainfall. And what you'll see is that, we've got the two methods. So this accuracy that I mentioned before. So this is the correlation between the predicted values for grain yield and actually the true simulated grain yield values in the data set for the method whole genome prediction without the network on the x-axis, and this APSIM WGP where we're exploiting these rules of biology on the y-axis. So the higher the value, the more concordant the results between the truth and the predictions. And what you'll notice is on this one-to-one line, this would be where they're performing similarly. What we're seeing is that including the APSIM sorghum model is giving us a predictive advantage over not including that in the genomic prediction model. And in pretty much all cases, apart from one, which is where we've trained, which I don't have time to go into today. The other piece of information underlying this graph is that the biggest improvement in accuracy, so the deviation from this one-to-one line, is predicting into trials or environments that we haven't seen before. So this is where the benefit of the crop model with this sort of pre-trained understanding of how genetics and the management and environments interact are able to adjust our estimates or predictions of the genetic component that drives yield. And this is sort of independent of the site we're looking at. So that was just a very quick demonstration of how that might help a metric that breeders might be used in, but something that I'm interested the more I get into it is, could these frameworks be used to go the other way? So how could we use this framework that can give you values of a position in the genome, a particular physiological trait, or the complex trait that might be selected on in a breeding program? How could those pieces of information be used in a different way? So one idea, back to early on with Christine mentioning gene editing and Charlie looking at targets in the genome, a lot of researchers would use something called a genome-wide association study to go after a complex trait, and they want to identify potential candidates to pass into an editing pipeline. So because we have this modeling capability from genes to the trait, there's nothing that stops us using the same information as a GWAS. So a secondary question, could we use this same framework, which is derived from the interests of breeders, to actually help learn more about the biology that we're testing in other areas of plant science. So just a very quick view. What I've got here is sort of an adjustment of a Manhattan plot. So you can think of the genome along the x-axis, so there's 10 chromosomes here. And I've zoomed into regions around where we've actually simulated the causal genomic effect in this virtual data set. So that's where these green ribbons are here. And what we've done is we've withheld those causal pieces of information from the prediction algorithm, and we've given them sort of 1,000 random bits in the genome to try and use as predictors of where those causal states might be. So in the orange, these are the predictions of the effect that would relate to a trait. So this was sort of phenology or maturity that you're looking at here. And we passed it just to put a caveat. This is a bit of a typo, actually. But we've got a very well measured plots for yield. So it's going to be a situation where you've got more genetic signal than noise, so maybe an optimistic scenario. But I think that just wanting to show you the potential, that what we could do is we can look and see that in 8 out of the 10 causal positions, we're actually identifying these SNP markers that breeders would use for prediction to identify sort of nearby positions in nearby QTL. And in most situations, this sort of central dot is pretty close to the predicted effect, pretty much overlaps with the true effect. And the only situations where we can't do that wasn't a problem with the algorithm, but it was actually because these two sites here, which aren't highlighted, were already fixed. So there's no variation, so we couldn't actually map them. So 2 points. I suppose we can look at a predicted effect and this uncertainty in the bars around that effect. So do we really want to go after this? And we're also not seeing false positives. So the areas outside of the green bands, you're not seeing orange dots jump out and give you false, sort of send you down false paths. So to conclude, I think we have set up a prediction framework that actually integrates AppSim, which has many crop models, to actually connect this genomic information and understanding of biology to breeding predictions. We've made a publicly available data set so other people in this space within or out with the center could look at algorithms they're interested in and compare against results that we're generating here. We've demonstrated in a virtual data set some improved prediction outcomes for sorghum. And that's both from going from the genome to the phenome, but also there's a potential to go from phenome back to the genome. This prediction platform is now operational on the national computing infrastructure. And although I've talked about sorghum here, the way we've set this up doesn't prevent any use to go after other crops. So if there's a model within AppSim that has a crop model, you could test this out if you had the appropriate data sets. And finally, I think to connect back through the last two talks, I think now we have a system in place with AppSim. It now changes the question from can we do this to what does the model need to be in order to go after the question we want. So I'll stop there and thank a lot of different collaborators and take any questions.
Thank you, Owen. Wasn't that great? Any questions? I'll ask one. We'll go to Tim. Okay, Tim? No. While he's going there, how soon before you'll try this out on a actual, like, agricultural, I don't know, some big data set that CSIRO has or somebody?
Yep. Well, I think we didn't need to look further than the centre as a start. So thankfully, we've got the team out at Hermitage. So we're having conversation with David Jordan and MMACE around developing appropriate data sets. It's not necessarily always that easy. So I think that's part of the ongoing conversation. So if we want to get down to understanding if we're getting predictions for the right reasons at physiological levels, that comes with a lot of intense field work that I'm sure Mark and Charlie can talk to when they did it in the private sector. But I think you can. I think where we are is we wanted to really validate the algorithm and how that's performing, and that lent itself to having a simulated data where we knew what the truth was. So we're in the position, we know it can rediscover the truth. I think changing those levels of heritability on the complex trait will give us some guidance around what crops are in a position to start using this in reality without sort of being able to be informed when they use it. So it's in process, but...
Yeah. So one of the problems, if I understand well, is if you've got big data sets like you're talking about, they're just not sitting there available in the public domain for us to sort of utilise, are they? They take a huge amount of resources to develop, so they're often protected within the company. But there are options, but it's also that they're probably developed for a different reason. So it means that you don't necessarily get the validation you need to carry forward. And it comes back to this identifiability. So you can still get good predictions of yield, but underneath the hood, you could be getting it for a variety of reasons. And I would suggest that people should do the investigations to know that level of uncertainty before they try to implement it commercially. Okay, Tim.
Oh, that was a great talk, and a very convincing demonstration of the value of crop growth models. I was talking to a rice person the other day at a conference, and I sort of was talking it up, and they just said, "Oh, well, we don't have a good crop growth model, and we don't have time to make one." I mean, how much of a limitation is that? It seemed like almost you guys are being very convincing. Is it just you haven't told everybody yet that they should be making crop growth models, or is the data just not good enough from those other crops?
I don't know. I think you talk to more people than I do. I think that was one of the reasons we started with sorghum, is the history and the development of that model, and in particular, connecting it to appropriate parameters. So this genetic to genotypic parameter within the model is really key to make sure that you're hooking something that's been modeled with genetics in mind. So I think that's the conversation to have. I'm probably not the best person to talk about crop growth model development, but certainly there are different groups outside of APSIM that are working on these things. So it'd really be going to people and say, are you in a position that you think it's ready? But I couldn't answer from my own expertise how to get them to that place.
You could certainly convince them that it's a good idea.
It's fair to say that, like Corteva has an in-house crop growth model, right? So a lot of those models may not be in the public domain either. It doesn't mean the concept of the linkage isn't really valuable. But does anyone in the audience want to comment on that?
Okay, yeah.
Ross?
I think there are partners within the centre that are exploring this beyond sorghum in other crops. So I don't think it's this is the only valuable demonstration. Probably stretches my own knowledge to say which crops would be good targets. Graham's ready to pounce. Yeah.
I can make a comment. Excuse me. Look, I think for most major agricultural crops, there would be models out there that are suitable to do this. They may not all be in APSIM, but they're certainly around. So it's usually just a matter of why not? Ignorance of the person who's making the remark.
Oh, so another thing. Well done, Tim, for talking it up. Because that's what I was talking about before. He knows what crop growth models are now, and I think that's something that we're sharing with this whole community. I think it's just beautiful. So, one more question was over here somewhere, Barbara.
Yeah, I just had a sort of technical, like when you have that nice picture up where you have genes on the left and they come into sort of APSIM, there's sort of this many-to-one map between genes and the components of the APSIM model. Is that sort of just how you've drawn it, or is that how you've sort of set it up? Or could it be like many-to-many with, I'm a canopy gene, but also I do weird stuff with roots just because of pleiotropy?
I could stand here for an hour and tell you why. I'm starting in yeast, but I won't. But that was just a depiction that Mark and I were working on, and how you would sort of move from the additive view. But in the sort of framework, there's nothing stopping you having those interactions beneath that level. It was just a nice way to show a progression from nothing interacting up to the complex stuff to sort of moving that back to something that might be more stable and more appropriate for that type of modelling.
all good.
All right, well, I think, really super quick. Oh, you want to wind it up?
Go, Charlie, yeah.
I love the research you did to go back to the signal detection, right? Would it be sensible to hypothesize that the ability to do that sort of backward identification depends on that linkage you have?
Yep, so it depends on the linkage. So I suppose underneath the hood you're specifying which parts of the model you want to look at and identify genetically. So we're not passing it to everything in the model simultaneously, but it's already incorporated in that framework that's iterating around. And I think the lower you go to where these interactions are less, contributing less, allows the model to deconvolute those interactions before you're going after these more consistent targets, which will give you a more sort of what we would like to see, which is we do this and it does that in many situations. Yeah.
How much computing power do you need? Do you need a Cray or something to run these models, or can you run it on your desktop?
Yeah, good question. So at the moment, I wouldn't recommend anyone running on the laptop. This is why we're using partners. So I had the NCI, National Computing Infrastructure. So as the center, we get access to their GADI high performance system. So we really need to exploit parallelization. So running the crop model independently in independent runs. So as an example, we're probably running these over thousands of different processes simultaneously to generate predictions in parallel to get good estimates of that uncertainty. So at the end, I just showed the bars, but what we actually have underneath is that full distribution of any potential yield that we've sampled across the trait space that underpins that bar that I showed at the end. So to get that sort of really detailed resolution, you need many simulations, because these are going to billions of predictions is what underpins even that small case study there. So well, I think this is the start, right? So I think that there's work around the software development. So that's where Graham's team with Jason and Greg come in. And we're in the development phase, and you can think there's also a productionization phase, which will be looking to improve that. Yeah.
We'll talk about it. We can continue on afterwards if that's all right. But thank you for your questions, everyone, and thank you for your answers.